And And we are recording this meeting, so we're hoping to share the link with you guys afterwards if you want it. I'm going to start by doing what I always do, which is introducing, well, first thanking the East End Programmers Association for working with us to make this possible. That's a consortium of the libraries of the East End who work together to bring more and better and many programs to all of the library patrons of the East End. I also want to welcome and thank Karen Wolfrad, who has been instrumental in getting this all set up, from Stony Brook Southampton. Karen has been there from the beginning, organizing programs for community health concerns for the library. So thank you, Karen, and I'm going to turn it over to you to introduce, do further introductions. So take it away. Thank you again.
Thank you, Nola. Yes, I'm Karen Wolfrad. I'm Director of Community Outreach, and I'm also the Administrative Director of the Regional Tick-Borne Disease Resource Center at Stony Brook Southampton Hospital. And this is another in a continuing series of new ways of getting information out to people. And please excuse our sometimes we don't know what we're doing here, but we've gotten through it so far. So hopefully this one will be just as successful as the other ones. It's my pleasure today to introduce Dr. Erin McGintee. And I'm going to read a short introduction so that you'll know about her background. She's also a member of our medical advisory panel for our Tick-Borne Disease Resource Center. Dr. Erin McGintee obtained her medical degree from SUNY Stony Brook and her training in pediatrics at Children's Hospital of Philadelphia. Following her residency, Dr. McGintee pursued subspecialty training during a 3-year fellowship in allergy and immunology with adult cross-training at the Hospital of the University of Pennsylvania. She's board certified in allergy and immunology. In 2009, she returned to New York and joined ENT and Allergy Associates, where she practices both adult and pediatric allergy and clinical immunology, with a special interest in the meat allergy Alphagal, which is caused by the Lone Star tick. She's a member of the medical advisory panel to the Regional Tick-Borne Disease Resource Center, and she has presented nationally on this subject and has been interviewed about Alphagal by a number of media outlets, including the Associated Press, Sirius XM Radio, and the BBC, among others. Dr. McGintee is also an appointed member of the Suffolk County, New York Board of Health Services, and she sees patients at her office in Southampton. Welcome, Dr. McGintee.
Hi, thank you. Thank you, Karen. Thank you, Nola. Thank you, everyone, for joining today. This is actually my first Zoom lecture, so I got over the first hurdle of would I be able to successfully screen share, and it was much easier than I thought it was going to be. So I am going to talk to you today about my particular interest and expertise in the world of ticks, which is Alphagal meat allergy. It's also called Alphagal syndrome. And I call this talk Midnight Anaphylaxis, Alphagal meat allergy from a Lone Star tick bite. I am going to try to give you some background on the history of Alphagal allergy. I'm going to give you examples of some cases. I'm going to give you a little background on the whole tick link, and then some practical information. And I hope to leave plenty of time at the end for any questions that you may have. I would ask that you keep your questions relevant to Alphagal allergy. I don't take care of Lyme disease or other tick-borne diseases. So while those are very important and are big public health issues out here, I can help you out with things relating to Lone Star ticks and meat allergy. Okay, so let's see now. Screen sharing is working, but how do I get, okay, there we go. So I'm going to present a couple of back-to-back cases of patients of mine who have been diagnosed with Alphagal allergy. So you'll see there are some similarities and some differences across the cases. So this first patient is a 31-year-old Latina female who gave a history of having had several tick bites over the summer, and she had also sustained larval Lone Star tick bites in August, and we're going to talk more about them later. So that was August. And at the end of August, this is 2018, she ate beef and pork tacos for lunch. 9 p.m. So, probably a good 8 hours later, she woke with abdominal pain and the urgent need to defecate. This progressed to a generalized hive rash, throat swelling, and difficulty swallowing. She took 2 Benadryl and then was not getting better, so went to the emergency room. And these were the test results on this patient. So Alphagal IgE, which is allergy antibody, was 4.2. It's supposed to be less than 0.1. Her total allergy antibody was 48, and that will become more relevant later. Case number 2, 83-year-old Caucasian male, no history of tick bites that he was aware of, though he did report that he had sustained chigger bites in August. And those are in quotes. I hope that most of you on the call here are aware that what people call chiggers out here are actually larval Lone Star ticks. So bites in August, early September, he ate a hamburger for lunch. And at 5 p.m., he noticed hives around his waist. He took some Benadryl and they went away. And I think at that point, he did not make an association that one had anything to do with the other. A couple weeks later, he had a steak for dinner, woke at 9 p.m. with, again, hives around his waist. But then when he got up to get out of bed, he had a brief LOC, that stands for loss of consciousness. So he actually stood up to get out of bed and briefly collapsed. Again, I guess nobody told him about alpha-gal allergy because he didn't make the connection that that's what was going on. On September 24th, he again had a burger for dinner and again woke up in the night with hives. And finally, after 3 episodes, he came to see me. His alpha-gal antibody, as you can see, was greater than 100, but his total allergy antibody was also quite elevated. So what is alpha-gal allergy? That's the first question. It is a blood group carbohydrate, which is a sugar that's expressed in all non-primate mammals. So if you're a mammal and you're not a human or an ape, alpha-gal will be expressed widely throughout all the tissues and cells. So cows, pigs, sheep, as you can see in the picture. But any mammal that's not a human or a monkey or an ape. So dogs, cats, squirrels, rabbits, they all have alpha-gal. So I think it's very interesting to give a little background on how was this allergy discovered? Because we've only been aware of this allergy for about 12 to 13 years at this point. And it actually was first identified in the world of oncology. And I can't take this slide out of my talk. I've been doing this talk for years. I just think it's too funny because, as most of you know, Martha Stewart is one of our local celebrities out here in the Hamptons. And she went to jail a number of years ago for insider trading. And that actually ties into the alpha-gal story, because the reason she went to jail was because she was selling her Imclone stock, because she had heard that this drug cetuximab that was being developed by Imclone was not going to get FDA approval. And it wasn't going to get FDA approval for this reason. It had passed all the clinical trials up until the phase 3 trials. And so you see these studies were published 2007, 2008. They started reporting in certain areas. One paper was out of Missouri, one paper was out of Tennessee and North Carolina, really high incidences of patients in the clinical trials getting this cancer drug and going into anaphylactic shock. So up until that point, they had only seen about 3% risk of allergic reactions to this drug, and most of them were mild. In these 2 trials, they were seeing upwards of 20% of the patients having these severe allergic reactions to this drug, which was highly unusual. So this is Bert O'Neill, who wrote one of those papers, and he thought it was strange too. So he started reaching out to oncologists in other parts of the country, and they thought he was nuts. He said down here on the bottom, they thought we were lying or crazy. So in places like Nashville, Missouri, they were seeing cases, and other parts of the country, they were not. This is a really complicated slide. I'm not going to go into the deep details of it, but basically what I want to show you is that they did a study and they were able to show that the patients in Tennessee who got cetuximab and were having allergic reactions already had blood antibodies that recognized cetuximab before they had ever even received their first infusion, as did other case studies. What's even more interesting is that they looked at other matched controls from Tennessee. So patients who didn't have cancer, who weren't going to get cetuximab, and a whole bunch of them also had antibodies that recognized cetuximab, even though they'd never seen the drug. So there was something that was making these patients make antibodies to cetuximab, even when they had never seen the drug. And when they looked at controls in California and in Boston, the vast majority of them were not showing any antibody to cetuximab. So there was definitely something regional going on. I just had to check if I had another slide here or if I have to tell you a little more. So interestingly, cetuximab was made in what they call a murine myeloma cell line. So it's a man-made antibody that they make in a cell line that has the enzyme that puts alpha-gal onto cells. So cetuximab, the drug itself, actually had alpha-gal on it. So there was something happening in the South where people were developing this allergy to alpha-gal without ever having had an exposure. So the next step was to figure out why the discrepancy. Why are these states in the Southeast seeing all of these cases where other parts of the country were not? And I'll tell you, in all these early studies, they didn't know about our little corner of the world and all the alpha-gal that we have up here. They know now, don't worry, they're well aware now, but most of this was a Southeast phenomenon when they were first looking at it. So how did they figure out that it was ticks that were causing this problem? It's actually also a very interesting story. So the first talk about how maybe tick bites could give you a meat allergy actually dates as far back as 1989, believe it or not, in a little town of Athens, Georgia. And there was a regular lady. She wasn't a doctor, Sandra Latimer, and she corresponded with Dr. Anthony Deutsch, who was an allergist in that area, because she was noticing people were having allergic reactions to meat that seemed to be happening after they had tick bites. So the two of them kind of looked into it. They ended up writing it up, and they sent letters in 1991. They sent letters to the Georgia Allergy Society and to the U.S. Center for Disease Control and Prevention, and it never got followed up on. So this was something that we figured out retrospectively. So some of the people who started doing alpha-gal research kind of had a little interest in, could there be something to do with ticks, just based on this initial report. In 2009, in Australia, which is right around the same time that we were discovering the tick link here, a doctor called Cheryl Van Noonen and her colleagues reported on 25 patients who were living in a tick endemic region of Sydney who developed red meat allergy after experiencing reactions to tick bites. So they proposed this as a hypothesis. My colleague Scott Cummins and his mentor Tom Platts Mills decided to dig into this theory and try to figure out if it fit with our cases of alpha-gal allergy. So I like this series of maps because it kind of makes a good point. So this is a map showing the 5 states that were reporting the high incidence of those allergic reactions to cetuximab. This is a map that way back in 2011 showed the states that were reporting the highest number of cases of allergic reactions to meat. So the states with the big stars are the ones that had at least 25 or more cases. So you can kind of see a trend here, right? This map shows the geographical incidence of Rocky Mountain spotted fever, a different tick-borne disease. So as you can see, 5 of the 6 states that were lit up in red in the first map are also lit up here. Tennessee and Mississippi are not that far behind. This was an old CDC map They haven't updated it. I'm sure the territory has expanded even further. But this was a map from 2012 showing the geographical distribution of the Lone Star tick population. I like to give a little caveat here, because when you look at this, it can seem really alarming. Yes, you could potentially find Lone Star ticks anywhere in this territory, but you have to remember, Lone Star ticks are only gonna thrive in an area that will perpetuate their life cycle. So if you look at this map, like all of Long Island is colored. My sister lives in Manhasset. They don't have deer wandering their yards in Manhasset. That's an essential part of the life cycle of the Lone Star tick. So you're not going to get a major raging Lone Star tick population in an area where there's no deer. So it's not to say that you shouldn't be worried about them in where we live, because as I'm sure any of you on this call know, we have a major deer problem. But there are areas, rural areas with rodents, birds, deer. Those are the areas where the Lone Star tick is really going to propagate and thrive. This is a little schematic that shows you the different types of ticks that we find here in the Hamptons. Okay, as you can see, the scale of this picture, there's an inch. This is a dime. Okay, so this is obviously extremely magnified. This is our black-legged tick, which is the deer tick. This is our Lone Star tick. And the important point I like to make here is a lot of people think if they had a tick on them, if it doesn't have a white dot, they think it's not the Lone Star tick, and they think that they're okay. A lot of people will say, oh, it was a deer tick. And I say, well, how do you know it was a deer tick? They say, oh, it was because it was really, really small. Well, this is the nymph deer tick. This is the nymph Lone Star tick. And this is the nymph dog tick. I didn't get to that yet, but you guys all know the dog tick. That was the old original tick here. This is a dime. So like, if you look at this schematic, you can see that they look a little different. But I don't know if anyone has good enough eyes to tell the difference between this and this, or even this, in real life without a magnifying glass or a microscope. So just because it's a tiny tick doesn't mean that it's not a Lone Star tick. Just because it doesn't have a dot doesn't mean it's not the Lone Star tick. This is the adult male. He does not have a dot. He's equally likely to induce alpha-gal allergy. A bite from any stage of the life cycle can trigger this allergy, okay? The final point I want to make on this slide is that this is our friend, the larva, that people call chiggers. Okay, we don't have chiggers on Long Island. I will debate anyone as long as you want to on that. When people are telling you you have chiggers, they don't understand you're being bit by larval Lone Star ticks, okay? And we know it's the larval Lone Star tick because that's the only larval form of the ticks in our area that will feed on a human host. So if you get those itchy bites on your legs, and I'm going to have a picture of them soon. I don't know if it's my next slide. Yes. In late August, yeah, it's actually even early August, you may see them. August, September is when these guys tend to get you. These are the larvae under a microscope, okay? These are larvae on someone's skin, but as you can tell, that's a super, super magnified picture. More often than not, the problem with the larva is you don't know you've gotten the bites until they've already bitten you and fallen off. These only, if they're undisturbed, they're only going to stay on you for like 4 hours, but you could almost brush them off with your hand and they'll fall right off. It's not like where you really have to pry them out like an adult tick. More often than not, you're going to have the rash, these itchy raised bumps, and some of them may get a little weepy and crusty, and they tend to be worse on the feet and the ankles because ticks come from the ground. You probably all know this, but I want to make sure, I don't want to assume anything. They're not going to fall out of a tree and land on you. You're going to step in an area where the tick is, and they're going to get on you from below, and they're going to work their way up. So when someone steps in a nest of the larva, the bites tend to be worst on the feet, the ankles, and the legs. It doesn't mean that a couple of them won't make it up further. You may get some bites in the armpit, in the groin area. but it does tend to follow that very characteristic distribution. They're not in you, just so you know, like they're not burrowed under your skin and they don't stay in you for weeks like some people think. They fall off. It's just an allergic response to the bite. And just because you have an allergic response to the bite does not mean you're going to get alpha-gal allergy. Everyone gets this skin reaction when they get these bites. So Scott, my friend Scott Cummins and his colleague began to suspect the ticks. So they looked at a couple of other things that further supported their hypothesis. One, they were able to show that patients who gave a history of reported tick or chigger bites, these are the red triangles, were more likely to have a higher alpha-gal allergy antibody, and they actually ran a higher total allergy antibody. Patients where they weren't sure whether they had had a tick bite or not, these are the black triangles, were less likely to be up here. And then patients who said, I know for a fact I've never had a bite, their total allergy antibody varied, but they had very little to no measurable alpha-gal antibody. So there was a clear correlation between people who specifically gave history of bites and the likelihood of having antibody to alpha-gal, okay? He then took sera, which is, you know, the white, the clear part of your blood, from 125 residents of Virginia and tested it both for allergy antibodies against alpha-gal and also tested for, they took Lone Star ticks and they like crushed them up and identified all the proteins, and they tested that those patients' sera also to see if they made allergy antibodies against the other proteins in the tick. And they also saw a correlation that the higher the alpha-gal IgE went, the higher the IgE to other tick proteins went. So another correlation. So neither of these little experiments were proof, but they both kind of supported their hypothesis. And then here, they actually were kind of able to convincingly demonstrate that tick bites led to elevations in both alpha-gal allergy antibody and total allergy antibody. So these are three patients, okay? And they're alpha-gal, this is just IgE, okay? The total IgE, the total allergy antibody, is marked for each patient in blue, and the alpha-gal IgE is marked for each patient in black, okay? And this is sequential samples over time for each of the three patients. So every time you see a red arrow on any of these three patients, that's a time when they say they got tick bites, a tick bite or multiple tick bites, okay? And so you can see on all three of them, after a tick bite, their total goes up and their alpha-gal goes up. The total goes up. This one, the alpha-gal didn't go up as dramatically, but it did go up. And you see it very clearly here, bite, and then they both go up. Then they start to go down, and then they get another bite, and they both go up again. The little asterisks, the little red stars, are actually points in the history where the patients actually had an allergic reaction to meat. So this one never did. But as you can see from these other two, they both had reactions at times when their alpha-gal antibody was peaking. So this was pretty compelling evidence that it was the bites from the ticks that were causing the changes in both the alpha-gal antibody and the total allergy antibody. So we still have a lot of other questions about ticks and alpha-gal allergy. Are tick bites the only trigger for the development of alpha-gal? And are Lone Star ticks the only one that can cause the alpha-gal antibody to go up in our area? Without getting into too much detail, we do think that in our area, the Lone Star ticks are the only ones that induce alpha-gal allergy, and we think that they're the only tick that will make your numbers go up. My colleagues down at UNC are looking at whether people who already have alpha-gal allergy, if they get stung by bees, could that transiently make their allergy worse? And that's something that they think might be a possibility. Now, they've not proven that a bee sting can cause alpha-gal allergy, only that if you already have alpha-gal allergy, it could possibly make it worse. They're looking at the mechanism, like how does this happen? There's some data that ticks have alpha-gal in their stomach and saliva that gets introduced in small amounts when they bite. And why is this continuing to go up? Is it because the ticks are expanding their territory and getting more aggressive? Is it because we're better at recognizing the condition? Or most likely a combination of both. So as I mentioned, in our area, the distribution and the analysis of tick salivary factors suggest that the Lone Star tick is the only cause of alpha-gal syndrome in the US. Having said that, this is not unique to our country. As I mentioned earlier, that there's a group in Australia that takes care of alpha-gal syndrome, and there have been pockets and cases reported in Europe, Scandinavia, Japan, South Africa, and there seems to be a different, they don't have the same ticks as us. So different tick species have been implicated as the cause of the allergy in these different regions. But here, the Lone Star tick seems to be the cause. The delay is very fascinating, and I think this is a very important thing to know about. So despite the fact that these patients have very high allergy antibodies binding to beef, pork, and lamb, they don't feel things immediately. If you talk to one of my patients who have peanut allergy, or talk to one of my patients who have shellfish allergy, one of the first symptoms of a food allergy is typically itching and discomfort in the mouth and throat. In fact, if a patient is concerned about a shrimp allergy and they don't have that, it makes me think it's very unlikely that that's what we're dealing with. But in alpha-gal, it's totally different. Patients could be incredibly allergic to meat, and when they're eating it in the moment, they don't feel anything, which is usually why they end up finishing a huge meat meal without realizing that they're going to react. The symptoms typically come on somewhere between 3 and 8 hours after, and I think you saw that in those examples I gave in the beginning. And it doesn't start in the mouth. The most common thing is itching and hives of the skin. I mentioned this, that there's this delay, and that is very unique to alpha-gal allergy. I think this is a cool little interview. This is from many years ago, July 2011. It's an interview with the author John Grisham. And if you look at this little part that's circled, they were asking him about his favorite food. And he said, I have this weird food allergy that started about 5 years ago, so I can't eat beef or pork, which is probably pretty healthy. We eat a lot of fish and fowl. I'd kill for a cheeseburger, though, or a big steak. So John Grisham, if any of you on this call have alpha-gal allergy, know you're in good company. This is some interesting pictures that I borrowed from my friend Scott when he was trying to prove that this delay was real. So these were patients of his who had reported that they were having these delayed reactions to meat, and they had positive testing for alpha-gal. And this was the lady before her challenge. And this is the picture of her 3 hours and 45 minutes after. And what they do for the challenges, and what I do for challenges in the office, is sausage patties, 2 sausage patties. I think this is probably projecting, but you can see her palms are extremely red and blotchy, and she's getting some redness up here as well. This was a patient, I can't read the top because I've got my screen sharing thing, but presumably there's some abnormal alpha-gal numbers up there on the top. And you can see this was the reaction. Those are hives all over this patient's arm, and that didn't start until 4 hours and 15 minutes after the sausage patties. And here's a third one, and I can read that one. So the alpha-gal antibody was 29, and remember I told you less than 0.1 is negative. The total allergy antibody was 201. Here's some hives that developed on this gentleman's side. And again, almost 4.5 hours after the challenge dose was given. Excuse me. So why the delay? Our theory is that it has to do with the fat. So in biochemistry, back when we're in medical school, we learned that fat is absorbed differently from protein or carbohydrates. So we typically start digesting protein and carbohydrates in saliva. We've got enzymes in saliva that start the digestive process there. Fat is different. Fat is relatively protected from any sort of digestion until it hits the bloodstream, 3 to 4 hours after you've eaten it. So it kind of makes sense that the alpha-gal that's bound to the fat part of the meat is not digested off. Like probably by the time the meat gets to the bloodstream, alpha-gal's been digested off of the protein part and the carbohydrate part, but the fat still has the alpha-gal bound to it because it's protected. What provides some support for this theory is the fact that fattier meats for sure cause more severe reactions than leaner meats. This is not debatable. This is an absolute fact. It's not to say someone with severe alpha-gal syndrome couldn't react to a leaner meat, but most of the bad reactions I see, it's a big portion of fatty meat. Another tricky thing about this allergy compared to other food allergies is that it's inconsistent, it's unpredictable. So fat content and portion size, as I just mentioned, are huge variables determining how likely it is that someone with this allergy is going to have a reaction and how severe the reaction will be. But alpha-gal, where their alpha-gal level is, which is directly correlated to the time since their last tick bite, is also really important. The higher your alpha-gal level is and the higher your alpha-gal antibody to total allergy antibody ratio is, the more reactive you're going to be. And that does change. You get a Lone Star tick bite and they go up, like you saw in the little figures a couple of slides ago. If you don't get any more Lone Star tick bites, they do eventually peak and start going down. So the worst reactions are when people are eating fatty meat in that window when the alpha-gal IgE and your alpha-gal to total ratio are very high. Much like other food allergies, we're discovering that there are a lot of cofactors that also may play a role in lowering the eliciting dose that might cause a reaction. So exercise after eating meat, alcohol consumption with meat, being on your menstrual cycle, being sick, taking certain medications like NSAIDs or aspirin, might make you more likely to have a severe reaction. And they're actually, I told you in the beginning, I don't know if anyone noticed, I told you there were going to be 3 cases that I presented. I don't know where one of my slides disappeared to, but the third case was a guy who had eaten like hamburgers and hot dogs at a beach party. And then he was drinking beers and he was surfing and playing beach volleyball. And he had a very severe reaction. And to me, it was a combination of the fatty meat, exercise, and alcohol kind of all together. And he had also had Lone Star tick bites a couple of weeks before that. So that's why it's not uncommon for me to see a patient who doesn't believe that they have this allergy, because even if they ate a hamburger and woke in the middle of the night with hives, they say, yeah, but I ate, bacon for breakfast, or I had a ham sandwich for lunch and I didn't have a problem. Just because you don't react every time you eat meat doesn't mean you don't have the allergy. So when I'm making a diagnosis of alpha-gal syndrome, it's really important that the history fits. And I'll get into that a little bit more later. But the reason this talk is called Midnight Anaphylaxis is because the vast majority of cases present in the middle of the night, which makes sense. People are most likely to eat their big meat meal for dinner, right? So 3 to 6 hours after dinner would be the middle of the night. It presents very similar to other food allergies in some ways, and there are some things that are different. So obviously the delay is different. The most common symptom we see is the skin, hives, flushing, itching. That's also the most common symptom we see in other food allergies. Gastrointestinal symptoms are very prevalent in patients with alpha-gal allergy. You can get gastrointestinal symptoms with any sort of food allergy reaction, but they're very common with this allergy. And a small subset of patients may present with only gastrointestinal symptoms and none of the other stuff. A little caveat there, though, is even the patients who present just with gastrointestinal symptoms, there's still acute reactions that happen 3 to 6 hours after eating meat. People with chronic abdominal pain, chronic diarrhea, unless there's a clear association that it's only happening 3 to 6 hours after eating meat, it's unlikely to be due to alpha-gal allergy. Moving on to the next one, syncope is a fancy doctor word for fainting. And pre-syncope is like when you feel like you're going to faint, you get lightheaded. So low blood pressure, dizziness, lightheadedness, fainting, not as common as the other two, but I see it often in the more severe alpha-gal reactions. And then less commonly, but some can also be seen are shortness of breath, chest tightness, wheezing, throat tightness, throat swelling. And again, not every single patient I see will give a report that they have had a Lone Star tick bite, but usually they will. And most often the bite will have happened in their recent memory. So you're most likely to have an alpha-gal reaction within a month of your last Lone Star tick bite, in my experience. I pop this up here just to show you that alpha-gal can be found in certain byproducts. I don't want you to get bogged down on this because the vast majority of these, there's not enough alpha-gal, there's either not enough in it to cause an actual reaction, or the molecule itself is too small to effectively cross-link your allergy antibodies on what we call your mast cells, which are the cells that release all the mediators of allergic reactions. So these are definitely exceptions rather than the rule. And on the next slide, I'm going to talk to you about what I believe to be the most relevant byproducts that you need to be aware of when you have this allergy. And there they are. Gelatin and cow's milk, by far, are the two most common byproducts that are implicated in this allergy. And even those are very rare. Okay, so that's the exception rather than the rule. So according to published data, less than 10% of alpha-gal syndrome patients are reported to be clinically reactive to gelatin or dairy. In my hands, I would say even less than that. And I see reactions more commonly to gelatin than to dairy. The vast majority of my patients, even patients who have had very severe meat reactions, are able to tolerate dairy. The reactions are generally dose dependent, meaning the more you eat or the fattier they are, the more likely you are to have a problem. This is something I actually only learned about fairly recently. Carrageenan is from seaweed, but it does have alpha-gal epitopes in it for some reason. I don't think I've ever seen a patient have a clinical reaction to it, but it has been reported in the literature. I do not, as a rule, tell all my alpha-gal patients that they have to avoid carrageenan. A lot of people worry about gelatin hidden in medications and things like that. So lots of vaccines contain a small amount of gelatin, but most of them have not been problems for patients with alpha-gal allergy. Exceptions are the MMR and the Zostavax, which is the old shingles vaccine. Rare, but there have been case reports of immediate anaphylaxis after injection of those vaccines. Fortunately, the new shingles vaccine, Shingrix, does not contain alpha-gal, so it's safe for alpha-gal patients to receive it. There are a few influenza vaccines that contain a small amount of gelatin. It does not seem to be an issue. The amount is insufficient to cause a problem in most alpha-gal patients. So look, if you have alpha-gal syndrome and you can get an influenza vaccine that doesn't contain gelatin, great. But if you didn't know and someone gave you one and they didn't realize it and had gelatin in it, it's highly unlikely to be an issue. I mentioned, liquid gels or gel caps. I have seen patients with alpha-gal allergy react to those. It's very rare. And what I will say to you is if you're someone with alpha-gal allergy recently diagnosed, if you take a vitamin D that's in like a liquid gel and you've been taking it all along, you haven't had a problem, You have to stop it just because you suddenly had a positive test. Like if you're taking it and tolerating it, it's just probably not enough to give you a problem. As I mentioned, the small amount of dairy, it's more common that people who react to dairy have gastrointestinal symptoms, and it can be very confusing because what's the most common issue that people have with dairy? Lactose intolerance, which is gastrointestinal. So I think lactose intolerance is far more common than alpha-gal reactions to dairy, but patients with a very high ratio between their alpha-gal antibody and their total allergy antibody, those are the ones in my experience that are at potential risk for dairy reactions. And as I mentioned before, as a rule, I don't tell all my alpha-gal patients that they need to avoid dairy, since upwards of 95% of them do just fine eating dairy. There are some medications that contain varying amounts of alpha-gal. Heparin contains alpha-gal, but generally it needs to be used in massive quantities to cause a problem. So like during like a cardiac bypass where someone's on like continuous heparin infusion, that might be a potential issue. But for the most part, the small amounts of heparin that are used to like Heplock and IV, not an issue. As you know, cetuximab, we talked about it, certain monoclonal antibodies are made in a cell line that puts alpha-gal onto the cells. There are a couple of others that have been reported rarely to cause problems. Armour thyroid is a natural thyroid medicine that's made from like pork thyroid. So some patients with alpha-gal syndrome may not tolerate that. And then the pancreatic enzymes are made from pork pancreas. Anti-venom, if you ever get bit by a snake, give me a call quickly. So I want to make sure I leave you guys plenty of time for questions. I identified my first case in October of 2011, so 2 years after I moved back out to the East End. And since then, I'm over at 750 cases. My case reports were the first reported on Long Island. Now I think lots of my colleagues, both my allergy colleagues and my primary care colleagues, have diagnosed many patients with the allergy. This is just I collaborated with Erin McGintee at NYU Langone at Winthrop and her fellow Ari Hefestoon, and we're writing a paper on my first 100 alpha-gal patients, and we're trying to get a sense of the incidence of patients presenting to an allergy office with a history of allergic reactions. And we basically did a retrospective chart review of all the patients who presented with a complaint of an allergic reaction or an anaphylaxis due to an unspecified food over a 10-year period. And then we graded the allergic reactions based on the history, and we calculated their alpha-gal numbers. Just real quickly, it was pretty evenly divided between males and females. Among patients who presented with those complaints of allergic reactions, 53% turned out to have alpha-gal. And that the time of symptom onset from ingestion was on average 262 minutes. And then you can see there was a huge range in the levels of patients who were positive, ranging from 0.22 to greater than 100, okay? And there was no difference between males and females, at least in terms of their alpha-gal IgE to total IgE ratio. The most common food in my food trigger was beef at 64%. These were the kind of symptoms, very consistent with what I told you. 80%, I can't see what that says, 80-something% urticaria, 82% rash in general. This is allergic swelling, abdominal cramping, diarrhea, shortness of breath, nausea, vomiting, and you can kind of see the others. Fainting, syncope, or low blood pressure happens, though much less likely than some of the other stuff. So our conclusions of this study were that about half of the cases presenting for anaphylaxis to an uncertain trigger were found to be due to alpha-gal syndrome in my office. Attribution to a true trigger is often complicated in these cases due to the delayed onset of symptoms, and that a good history is key to diagnosis. The reaction characteristics in my case series are similar to those reported in other regions. And our findings suggest that combining clinical history with the Alphagal to total IgE ratio is a more generalizable diagnostic tool compared to the use of Alphagal IgE alone. And that is very important. Because sensitization does not always mean clinical allergy, okay? So having a positive Alphagal IgE test does not always mean that you're gonna have a reaction to meat. They've done a couple of small studies, and I'd like to do one in my area as well. They've taken groups of patients in areas that are endemic for the Lone Star tick and just measured Alphagal numbers in all comers. And depending on which study you look at, the level of sensitization, meaning the percentage of people who have a positive Alphagal IgE test, ranged somewhere between 15% to 35%, so not insignificant. In those same studies, actual clinical reactions to meat only happened in somewhere between 1% and 8% of sensitized individuals. So the take-home point here, and I try to get this message out to as many people as possible. I know it's scary and nerve-wracking when you get a Lone Star tick bite and you're scared you're going to get this allergy, but you're much more likely to have a positive test than you are to actually have the allergy. So I do really try to discourage patients and doctors from testing everyone for Alphagal syndrome the minute they get a Lone Star tick bite. So in summary, evidence suggests that tick bites can affect both your total allergy antibody and your specific allergy antibody to Alphagal in some patients. In many cases, this sensitization appears to decrease over time. I would say pretty much in all cases. And additional tick bites may lead to it going back up. And that's it. And hopefully I left some time to address some of these questions. Thank you for your attention. Let me try to get to the top. Why do the southern states have the Lone Star tick? No, we all have the whole southeast straight up to Maine, all where that, I can probably pull the map up again. Let me. And while I'm pulling this up, let me see. So this is the CDC data territory where you would expect to find the Lone Star tick. This map hasn't been updated. It might have even spread a little bit more. But that's where we are right now. Where did my chat box go? Here it is. All right, let me get back up here. What is a high antibody number, and when do you know it drops? Blood work, I have headaches and fatigue. Yeah, so headaches and fatigue, we don't think that has anything to do with Alphagal syndrome. Obviously, there are other tick-borne illnesses that can cause headaches and fatigue, and if you're someone who's prone to getting tick bites, you could have other tick-borne illnesses, but also headache and fatigue, as a physician, I can tell you, is a very common and very non-specific finding, and it could mean a whole bunch of other things. So I wouldn't probably put a lot of weight on headache and fatigue as being related to Alphagal, especially if you're not eating meat, okay? And again, if you're avoiding meat and you're having vague and unusual symptoms, it's probably much less likely to be that you're eating some rare byproduct that you're missing, and it's probably more likely that you just have something else going on. But to answer your question about what is a high antibody number, as you could see from my data, it can range very, very broadly. So I had patients having allergic reactions to meat whose antibodies were 0.22. And I've had others who have not reacted to meat whose antibodies are greater than 100. So you really have to take the clinical history into consideration, okay? The type of reaction the patient had and how compelling it is that it was an alpha-gal reaction. But you also, I made this point, but I'm going to make it again. I can't look at an alpha-gal IgE number alone. I have to look at the total with it. The ratio of alpha-gal antibody to total allergy antibody is much more helpful than the number alone. So we use this cutoff that's not really been established in studies yet. It's sort of a kind of clinical experience thing. We use a cutoff of 5%. Doesn't mean that if your ratio is higher than 5% that you maybe couldn't tolerate some meat, but in general, we use a cutoff of 5% before we talk about potentially allowing a patient to reintroduce meat into their diet. And I check people's numbers, depends, generally twice a year. I like to check them once right before tick season, and I know you can get a tick anytime, but the major tick season. So like early spring, I like to check it, and then I like to check it at the end of fall. If somebody's numbers are improving really quickly, maybe I'll check it a little more frequently. If somebody's numbers are going down very, very slowly, and especially if they're not super eager to eat meat, we may wait a year to test them. Is there a particular gene that makes, we don't know. That's a really good question. We don't know, but what I will tell you is that once you have it, and I think this is what, I think you know this already, or you wouldn't have asked this question. It's not like certain Lone Star ticks carry it and other Lone Star ticks don't. If you have the ability to make alpha-gal antibody, and we don't know why some people do and other people don't, any Lone Star tick is going to make the numbers go up. And your second question, does the syndrome ever go away? It can, if you can go long enough without getting Lone Star tick bites. The numbers predictably trend down the longer you can go without getting a bite. It's hard for me to tell you that that's 100% for everybody, because every once in a while I see someone and they swear to me they haven't had a tick bite since their last test and their numbers went up. What I tell them is the only thing we know that makes the numbers go up is a new Lone Star tick bite. So theoretically, if you move to Alaska or somewhere where there's no Lone Star ticks, over time it would go away. This question might be further down, but in terms of how quickly it goes away, it varies from person to person. It really does. So you can't say, oh, my neighbor told me in 6 months it'll go away. I hear that all the time. Or my doctor said, give it 6 months, it'll go away. It really can vary from person to person. So you can't generalize about that. This question, Betsy, that's what I get for trying to be silly. Yeah, anti-venom has alpha-gal in it. So like if you ever got bitten by a snake, look, it's a risk-benefit ratio. We also don't want you to die from the venom. And I don't know, we don't fortunately have venomous snakes here to my knowledge, but if you spend time in an area where there are venomous snakes, there can be small amounts of alpha-gal in anti-venom, but if you get bitten by a snake that's going to kill you, you get the anti-venom, and if you're having allergic symptoms, then they treat that secondarily. It's just good to be aware that there's a potential risk for reaction. Are there drugs being developed? Yeah, I mean, I know that there is a lot of ongoing research. They're looking at a monoclonal antibody called Xolair that we use to treat chronic hives and asthma. It's an anti-IgE monoclonal antibody. It's like ridiculously expensive. So I think we'd have a hard time compelling insurance companies to pay for it to help with an allergy that really can be avoided by not eating meat. But Scott Cummins did a really small scale study where he showed that, I think he looked at like 8 patients, that Xolair was helpful. There's a lot of ongoing research in developing gal-free lines of mammals. So there's a gal-free pig that's genetically engineered that it just doesn't have alpha-gal. and it was developed for transplants because alpha-gal is also a major cause of why humans reject transplants from non-primate mammals. But they're looking down in North Carolina at doing challenges with alpha-gal patients with the gal-free pigs. And the alpha-gal patients seem to tolerate the gal-free pigs. I'm not aware of any other therapeutics that are ready to hit the market, but I do have a colleague who works in pharma who has been trying to coordinate with me to get access to sera on my alpha-gal patients. So I know that some of the pharmaceutical companies are exploring that as a possibility, but I don't anticipate it happening anytime soon. Yeah, this question, I think I answered. The average time between alpha-gal allergy and when we can eat meat again, can't predict that. To way too many variables. It depends how high your level is, how high your ratio is, how severe your reaction was, and if you get more tick bites. I've had patients eating meat again in a year, and I've had patients who just never can seem to get low enough because they just keep getting tick bites. And the other thing to remember is it's not even that simple, because some patients can eat some meat at some times. Like I have some patients who can eat lean meats, especially in the winter when they haven't had any recent tick bites, but they can't eat a hamburger or a platter of ribs, you know. So it's not black and white. There's a lot of gray area, and that's why it's helpful when you're diagnosed with this allergy to be followed by an allergist who can kind of guide you in that stuff. Do I see cases where IgE test is I have seen where IgE test is negative, but clinical presentation leads to diagnosis. In general, it's a very reliable test, so it is very unlikely for someone to have alpha-gal allergy and not have alpha-gal IgE. I will say that there's probably a couple of caveats. I mean, I've seen it 2 or 3 times where patient sounds very, very suspicious for alpha-gal and their test is negative. So one possibility is they had it, and from when they had their reaction to when we did the test, the antibody went away, you know, that they had it very transiently. Like they were that person, that second person of those 3 figures, the one where their alpha-gal went up a tiny bit and then it went back down. And I said that one never had a reaction. If that person maybe had eaten meat right when it was peaking, maybe they would have had a reaction. And by the time someone tested them, the test might have been negative. There's also the theoretical possibility that someone is, again, on the downswing from alpha-gal. So they have some alpha-gal antibody, but it's all bound already to their allergy cells, ready to react. And there's not enough in the circulation to measure alpha-gal on a blood test, but it is very, very unlikely. So if you figure I have 750 something patients with the allergy, I've in 10 years, I've seen maybe 3 where I'm like, wow, this really sounds like alpha-gal allergy, but the testing comes up negative. So it's uncommon. Usually the test is very reliable. How long does the tick need to be on you before you become infected? So just a matter of semantics. It's not really an infection. It's just an allergy. It'd be a completely healthy newborn naive tick with no diseases, but does not have to be on there long. It's not like Lyme or Ehrlichia where we really think the tick needs to be feeding for a while. I mean, the larvae often are on for like an hour or less, and they can still induce this allergy. So not long. If it's attached and you already have alpha-gal syndrome and a new Lone Star tick bite, a new Lone Star tick is attached, you're probably universal glad. There is no correlation between shellfish allergy and alpha-gal. In fact, there's no correlation between alpha-gal and any other allergy. You could be a super allergic person with tons of other food allergies and environmental allergies and get alpha-gal, or you could be a person with no other allergies and get alpha-gal. Yes, if I tested positive, did you say I might not be allergic to meat? Yes, you can be sensitized and not necessarily be clinically reactive. That's why I always tell people, like, it's not ideal to just test. And I know that's a scary idea that, oh, I got bitten by a Lone Star tick. What am I just supposed to go eat a hamburger and hope I don't go into anaphylactic shock? And I say, no, you shouldn't do that either. But if I tested everyone who got a Lone Star tick bite, I'd find a lot more positive tests than people who are actually reactive. So when I get a Lone Star tick bite, I unfortunately haven't had one in quite a few years, but when I get a bite, I don't test myself. But what I do is I'm cautious with meat for about a month. I'm not saying I don't eat it, but I don't eat a giant hamburger from Rowdy Hall in East Hampton like 2 weeks after I had a Lone Star tick bite. Remember what I said, fattier meats, bigger portions, worse reactions. Most people with this allergy, even when they have reactions, do not have full-blown anaphylactic shock. I'm not saying it can't happen, but that's the minority. And that almost always is associated with eating a big portion of meat within a couple of weeks of a Lone Star tick bite. So I think a prudent approach when you've had a Lone Star tick bite is be cautious with fatty meats for about a month. Stick to lean meats. If you feel anything, upset stomach, some hives, then you stop, you take Benadryl, you get tested. Everyone who has alpha-gal allergy that I take care of gets a prescription for an EpiPen, but I'd say no more than 5 to 10% of them have ever needed to use it. So anaphylaxis with alpha-gal allergy is the exception rather than the rule. Oh, I think this is a follow-up question about the map. Yeah, so this one shows the distribution of Rocky Mountain spotted fever, which is caused by a different tick, but the point of it was just areas endemic for ticks, that they all kind of followed the same pattern on the map, and that's sort of what raised their curiosity about whether or not ticks could be causing this. Is it possible that eating dairy leads to immune system stress over time, even if intense symptoms are not present? That's a really good question, actually. We don't have any evidence to show that, but they are wondering whether patients with alpha-gal antibodies and even alpha-gal antibodies who are not meat reactive, if eating it day in and day out could be promoting like micro inflammation. Not been proven. It's the hypothesis that they're wondering about. So there's certainly no harm in avoiding dairy when you have the allergy, but it's a big ask for a lot of people, especially when we don't really have any compelling evidence to say for sure that it's a problem in the vast majority of people. But that's a really good question. Okay. This question says, had a general tick panel without alpha-gal, test positive for Rocky Mountain spotted fever, no symptoms. Husband tested positive for alpha-gal. You don't eat red meat anyway, put on doxy, told to continue to not eat red meat. Should I get the alpha-gal test anyway and just pay for it? Okay, so I'm not speaking about Rocky Mountain spotted fever, but I am going to speak about it really briefly just because I see this all the time. Rocky Mountain spotted fever is not subtle. You can't have asymptomatic Rocky Mountain spotted fever. People get false positives on that test all the time, probably because of cross-reactivity with some other rickettsial bacteria in ticks. So your husband, it sounds like your husband had a test without symptoms, so certainly could be false positive. And my answer to should you get the alpha-gal test, I think I kind of just answered that. I mean, I'm not in favor of doing it just to do it, because I think you can get false positives. And if you don't eat red meat, I wouldn't worry about it. I don't know why they told you to not eat red meat when you didn't even have an alpha-gal test. If you're really anxious about it, you could certainly get the test done. But if you don't eat red meat, honestly, I wouldn't worry about it. I just wouldn't worry about it. I have time for a few more. Eventually, I'm gonna have to go because I do have to see patients. Here's another anti-venom question. I think I answered it. Yeah, I mean, yeah, I think you have to weigh the risks and the benefits. There's no guarantee that you're gonna react to anti-venom just because you have alpha-gal allergy. It's just to say that there is a little bit of alpha-gal in it. And I think if it's a life-threatening paralytic venom bite, then yeah, you should just take the anti-venom. Told to get I carry an EpiPen for bee stings. Husband was told to get an EpiPen because he is positive for alpha-gal, so that's on order. Yeah, I mean, I don't know if your husband just has a positive test or if he, unless I was the one you just asked. Yeah, yeah, I mean, I give everyone, once they have a positive test, even if your husband's never reacted to meat, and it certainly could be a false positive, it's like once that can of worms is opened, yeah, I mean, I think it would medical legally, it would be foolish to not give him one. But I really doubt he's gonna need to use it. Alphagal, take armor thyroid. No, your armor thyroid won't make, I'm sorry if I'm not reading the questions. I have Alphagal, I take armor thyroid, I'm thinking now my numbers will never go down. No, that's a good point. Eating meat, taking things with Alphagal in them will not affect your numbers. The only thing that will affect your numbers is a new Lone Star tick bite. So if you're taking armor thyroid and it's not bothering you, it's not gonna have any impact on your numbers. The only thing that will affect your numbers is Lone Star tick bites, okay? Oh, thank you for such a great information and beautiful presentation. You're welcome, thank you. If Lone Star nymph bite led to no detection of any diseases on it. Yeah, no, because on a standard tick paddle, is it safe to eat meat or is there a chance of false negative result? The Alphagal test wouldn't be on a standard tick panel unless they specifically tested you for it. And the question is it safe to eat meat? I mean, we don't know. You know what I mean? I would do what I told you I'd do. I would just try small portions of lean meat and see how you feel. And if you're feeling any symptoms, then you get tested. And if you're not, then after a few weeks, you kind of can gradually increase. Can we sign up for trials or research? Right now, I am not doing any clinical trials, but for sure, if there is any clinical trials which I hope to do at some point in the future, Karen and the Tick-Borne Disease Resource Center will probably make that information available to the public, and I would be happy to have anyone who's willing to be involved. Thank you. You're welcome. You're welcome. You're welcome to everybody. Thank you for all the thank yous.
Thank you, doctor.
I think that's all the questions for now. And if anyone is not my patient and you want to come and see me and talk about it more, I'm happy to. I also do telehealths on Friday mornings, which is a great, especially if people live on the North Fork, especially in the summer. It's really easy to do, to evaluate Alphagal over telehealth because it's it's not really something I had to do a laying of hands. It's more like a history and a blood test. So I'm happy to see any of you at any point.
Okay, then I think that wraps it up. Thank you so much. This has been so helpful and so informative and so clear. We really appreciate it. And,
it was my pleasure. It was my pleasure. Thank you for, I'm thrilled that there's been such a great turnout, and thanks for all the fantastic questions. You brought up some stuff that I didn't mention in the question, so that was really great.
Well, we really appreciate it, all of us, on behalf of all the libraries of the East End. Thank you.
Awesome.
Thank you.
My
Thank
pleasure.
you.
Take care, everyone. Have
Thank
a
you.
tick-free summer.
Yeah, really. Bye. Thanks a lot.
Bye, Karen. Bye.